Hello, my dear students. This is very important topic. You can short for CBSE, which we are doing for the exam purpose. So, structure of the microsporangium and microsporogenesis. You can see here in the diagram. This diagram is from your textbook. So, there are when we talk about the TS, that is transverse section of your anther, there are four layers. Epidermis is there, endothecium is there, middle layer are there, and then tapetum is there. So, if you look at this diagram, you have epidermis here, then endothecium, then there are more middle layers. That means it's not a single layer. There are many middle layers. Then you have tapetum is the innermost layer. Innermost layer, it is nutritive. Tapetum is nutritive. It is binucleate. Why is it binucleate or multinucleate? Because it needs to provide more and more nutrition for the developing these microspores. Then you have sporogenous tissue, which is inside, and then it is present at the center. It is compact. There are homogeneous cells, and each cell is a potent PMC, that is pollen mother cell. Then what happens in the microsporogenesis? Pollen mother cell undergoes meiosis. It forms microspore tetrad, four microspores. Like when the meiosis will happen, there will be four microspores formed, and each microspore is haploid. Then after that pollen formation happens, microspores, they separate from each other, they develop into the pollen grains. They are released from the anther by the process of dehiscence. So you see these lobes of the anther, they are opening at these places. So this is called a dehiscence line. So from here, these microspores, what I mean to say, pollen grains, they will be released out. Now, what is the difference between microspore and the pollen grain? So microspores are immature pollen grains itself. These microspores, they are bound together. They are held together by a thick wall, which is called as callus. It's an extra information for you. It's called callus. So then after some time, and enzyme, when these microspores have to mature to turn into the pollen grains, there's an enzyme which is called as callase, 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 which dissolves this callus wall. This callus wall is actually polysaccharide. It's made up of polysaccharide. So you have callose, which keeps these microspores together, and then you have callase, CLLASE. Callase is an enzyme which dissolves this callose layer, and this callose dries up, and then these microspores are turned into the pollen grains, mature pollen grains, and then dehiscence happens here. Then you have these two questions which are given in your textbook. How do tapetal cells become binucleate? By mitosis without cytokinesis and or fusion of the nuclei. So cytokinesis will not happen, nuclear division will happen. That's why many nuclei are present in the tapetal cells, and ploidy of the cells in the microspore tetrad is always haploid. Just remember these things. Now next lecture will be about something else. So hope you're enjoying these lectures. Thank you.